Today we are going to talk about DNA and RNA. You already know about DNA. You need your notebook. We're going to be doing notes. Here's my really boring title slide because I didn't put any pictures on it. But this is what we'll be going over today. All right, here's where we're at with our learning targets, okay? You should already be able to give examples of proteins and describe those functions, okay? Compare and contrast the structure of DNA and RNA. That's what we're looking at today, okay? So your next assessment will be over these 4 learning targets. And today specifically, we're gonna look at DNA and RNA. All right, I want you to take a picture or take a second and look at this picture. You don't have to write anything down here, okay? So the image shows DNA and RNA. I want you to find 2 differences between DNA and RNA. So now that we've looked at 2 differences, you've picked them out, we're gonna write down a bunch of the differences, okay? So DNA is double-stranded, meaning it has 2 strings or 2 strands or 2 sides, okay? So we can see here, here's one strand, right? A strand is like a side or a string, and here's the other strand. Remember we said it's a double helix because it's got 2 strands. RNA, on the other hand, is single-stranded, meaning it's only got 1 strand or 1 side, okay? Now, they're both twisted like a helix, right? But one's a double helix and one's a single helix. DNA has deoxyribose as its sugar, okay? Which is easy to remember because DNA starts with D and deoxyribose starts with D. Why do we call it deoxyribose? Well, if we take a look at this sugar, remember we know that the sugar is the pentagon shape, okay? With this sugar, it's deoxy. It's missing an oxygen. So notice here we only have an H. We don't have an OH, an oxygen and a hydrogen. In RNA, we have a sugar called ribose, which is easy to remember because ribose and RNA both start with R. And the reason we call it ribose, notice ribose is in deoxyribose, okay? Ribose has that oxygen. It's not deoxy, right? Deoxygen, not oxygen. So ribose has the oxygen, deoxyribose does not. You do not need to know that for the test, the oxygen part. You do not need to know the difference in the structure between these 2. You just need to know the names, okay? What's written in blue and pink. DNA has 4 bases, A, T, C, and G. You should remember that, right? Adenine, thymine, cytosine, and guanine, okay? RNA does not have T. Okay, it doesn't have thymine. Instead of T, it has a U. So it has A, C, and G as well, but it doesn't have the T. It has U, and U stands for uracil. Now, why does it have a different letter? I have no idea. Has to do with evolution, okay? But when we're looking at the differences, just know RNA has U, DNA has T. DNA can't leave the nucleus, okay? So DNA is stuck there. Remember, DNA has the instructions to make proteins. So DNA is super, super important. We want it to stay in that nucleus where it's kind of protected, away from other stuff. It can't get damaged, right? Remember, damaged DNA is called a mutation, right? A change in our DNA. RNA can leave the nucleus. So RNA can go in the nucleus, it can go out of the nucleus, it can go wherever it wants. So those are the differences between DNA and RNA. You might be asking, Ms. Frankson, why are we talking about RNA? Well, RNA is going to be really important for helping us to make proteins, and that's going to be our next topic. How do we make proteins? We know DNA has the instructions for making proteins, but how do we actually make proteins? So we're going to be looking at RNA and how it's used to build proteins.